Good morning, learners. My name is Mr. Pochilola once again. This is Black Slides Online Secondary School, the platform where you learn the sciences. The sciences being offered at MSCE. We continue with our revision sessions. Examinations are just very near now, much nearer than we have been thinking and talking. Exams are starting in 8 days' time. Here at Black Slides, we are not going to get tired of preparing you for the exams. Welcome, learners. If you are watching us on our YouTube channel, which is Black Slides Online Secondary School, consider subscribing to the channel. If you have not done so already, consider subscribing to the channel so that every time we upload a new video lesson, you become informed on time. Welcome, learners. We continue with our revisions. Invite your friends to come to join us to the platform to revise with us. We are going to be looking at a paper, Biology Paper 1 from Karonga Diocese. I consider this paper to be one of the best papers I've seen this season. This is a paper from Karonga Diocese. Karonga Diocese has some of the best schools in the country, namely Chaminade Secondary School, Kasei Girls Secondary School, St. Mary's Karonga Secondary School. These are schools in the diocese, Catholic Diocese of Karonga. They are high-performing schools. It's in this same diocese that you find schools such as St. Ignatius of Loyola Secondary School. It is in the same diocese where you find schools such as St. Mauritius Secondary School, but you also find the CDSSs there, such as Ngara CDSS, Furirwa Secondary School, St. Anne's CDSS, and St. Monica. These are Catholic schools that are doing very well in the diocese, and of course in the countries. They are among the best performing schools. So it's our privilege that we have laid our hand on this paper. I encourage you that you should watch this paper. You should learn the tips, the exam tips, as I bring them forth to you up to the end. Invite your friends. Welcome, learners. Remember, here at Black Slides Online Secondary School, our intention is not just to teach, but to prepare you fully for the MSCE examinations so that you come back with the best results that you can get. In this case, we are looking forward to having you get as many distinctions as possible. Welcome, learners. Don't hesitate to contact us on the numbers that we have shared before. In case you have you want to make inquiries regarding our school, you are welcome. Or you can simply leave a comment in the comment section. Then we'll be glad to address any matters raised there. Welcome, learners. Invite your friends wherever they may be. This is Biology Paper 1 from Karonga Diocese. Welcome, learners. Subscribe to the channel. Invite your friends. Hit the notification bell so that every time we upload a new video lesson, you are notified on time. This is Black Slides Online Secondary School. Without further delay, let's go on. Let's get to the business of the day today. We are revising the Biology Paper 1 from Karonga Diocese. Which consists of schools such as Chaminade Secondary School, Kasai Girls Secondary School, St. Mary's Karonga Secondary School, St. Ignatius of Loyola Secondary School, St. Mauritius Secondary School, and some CDSS schools, namely Ngara CDSS, Fulirwa CDSS, St. Anne's CDSS, and St. Monica, among a few. Just to mention a few. There are more schools than these which I've mentioned now. Welcome to section A. Answer all the questions in this section in the spaces provided. Figure 1 is a diagram showing a human cell undergoing cell division, a cell division. Use it to answer the questions that follow. Just a quick look at the diagram. The diagram is showing homologous chromosomes pairing up. Still in the nucleus, see? It's still showing the nuclear envelope containing chromosomes. And from the look of things, the cell is undergoing some changes. It's undergoing a cell division process. So the question is, identify the type of cell division that is presented here. Remember, we have two types of cell divisions, namely meiosis and mitosis. Meiosis is a cell division involving sex cells, such as the sperms and the eggs, in the formation of offsprings. And mitosis involves the multiplication of ordinary cells, such as the body cells, popularly known as somatic cells. Welcome, learners. So identify the type of cell division that is presented here. This is a division called meiosis, meiosis 1. And it's not mitosis. From the diagram, give a reason for your answer. The reason must come from the diagram. So why we think it's meiosis and not mitosis is because it shows that the homologous chromosomes, identical chromosomes, are paired up. They're paired up together. We have these two identical chromosomes pairing up. These two identical chromosomes also pairing up to form what are called the tetrads, or sometimes known as bivariants. The pairing up of the homologous chromosomes is testament to the fact that this must be meiosis and not mitosis, because meiosis involves chromosomes, homologous chromosomes pairing up together. Which only happens in meiosis and not mitosis. And the number of chromosome sets is being reduced. Also, the number of chromosome pairs, chromosome sets, is being reduced, a key feature of meiosis. So these are reasons why we consider this to be meiosis and not mitosis. At what stage of cell division is the cell? Give a reason for your answer. This stage of cell division, or the stage of meiosis, is called the prophase 1. The reason we think that is prophase 1 is because the chromosomes have condensed and become visible as distinct structures. So we are able to see the structures clearly. Chromosomal structures, which only happens at prophase 1. Let me encourage you to look at the other stages of meiosis, such as anaphase, metaphase. This is just the prophase 1. So we are saying we are confident that it's prophase 1 because chromosomes have condensed. They are distinct structures that we can see. They are visible structures, these chromosomes. We are also sure that it's prophase 1 because the homologous chromosomes, homologous chromosomes, these ones, are paired up. So the pairing up can only happen at prophase 1 of the meiosis cell division, meiotic cell division. This is the process called synapsis. So synapsis has occurred. The pairing up of the homologous chromosomes is called synapsis. It has happened. That's why we are sure this is prophase 1. Another reason we consider this to be prophase 1 is because the nuclear envelope is this thing here. It is still present and not yet broken. And the spindle formation has just started. These are the evidence that this must be prophase 1. Like I said, please revise the other stages of meiosis, just to be sure, in case the examiners may decide to give you that as part of your exam come July. Another proof or evidence that this must be prophase 1 is there is crossing over. Crossing over, you see this blue chromosome and this red one, they are crossing their chromatids here. So crossing over is also occurring between the homologous chromosomes. It's visible as the segments of the chromatids are overlapping. Here they're overlapping. It shows that there is crossing over there. The genetic material from this chromosome and this one are blending there. In order to determine the traits of the offspring from mother and father. State two functions of DNA in the body. DNA is short for deoxyribonucleic acid, which is a protein molecule responsible for protein synthesis, giving instructions for the synthesis of proteins in the body. All right, so state two functions of DNA in the body. One of the functions of DNA is that it stores and carries genetic information. It's the blueprint for genetic information of offspring. So it stores and carries genetic information, the instructions for the development, functioning, growth, and reproduction of all living things. It's DNA, that's storage of genetic information. By just saying it stores and carries genetic information, that's one mark. Or it stores and carries instructions for the development, functioning, growth, and reproduction of all living organisms. One mark. It passes genetic information from parents to offsprings, allowing genetic information to be inherited. So it passes genetic information from parents to offspring. It's the DNA is the one that carries such information, allowing genetic information to be inherited or to be passed on from parents to the offspring, children. It also provides the code for making proteins, which controls cell activities and traits or characteristics. So it stores and carries genetic information. Passes genetic information from parents to offspring. It provides the code for making proteins. Any two of these 3 points, 2 marks. Don't write all of them. After all, the question is just 2 marks. Welcome, learners. My name is Mr. Paul Chilola, your teacher. Number 2, explain how the liver ensures a steady supply of glucose in the body. Explain how the liver ensures a steady supply of glucose in the body. Remember that one of the functions of the liver is to regulate sugar or glucose in the body. Carries out a number of functions. One of them is to regulate sugar in the body. Another one is to produce bile salts. Another one is to carry out deamination of excess proteins. It also detoxifies poisons in the body, harmful chemicals. But here we are asked to explain how the liver ensures steady supply of glucose in the body. In other words, we are looking at the regulatory function of glucose done by the liver. So what the liver does is that when blood glucose level is high, The liver converts excess glucose to glycogen and stores it. Remember that glycogen is the form in which the body stores carbohydrate. It's a carbohydrate, it's a long chain carbohydrate, just like starch in plants. But here we are looking at humans. Glycogen is a carbohydrate, it's a large molecule, it's a large long chain. It's a polymer, in other words, whose monomers is glucose. So the liver converts excess glucose into glycogen and stores it. Welcome, learners. So when blood glucose level is high, the liver is going to change to convert that high amount of glucose, the excess glucose, into a form that the body can store. And the storage facility is the liver itself. On the other hand, when blood glucose level drops, like is low, the liver breaks down the stored glycogen into glucose and releases it into the blood, keeping the levels stable. 2 marks for explaining all this. State 3 things that could happen to amino acids absorbed from the gut. Remember, amino acids are the final products of protein digestion. When the body digests proteins, the final product is the amino acids. It's the building blocks of proteins. So the question is saying state 3 things that could happen to amino acids absorbed from the gut. Gut is the digestive system. Digestive system is also called the gut, or also known as the alimentary canal. In fact, gut means alimentary canal, which is the channel through which food moves as it's being digested. So, what is the fate? Some questions would say state the 3 fates of amino acids in the digestive system. Where do the amino acids go? When proteins are digested to form amino acids, where do the amino acids go? What happens? That's what this question is finding out from us. So one of the fates of the amino acids is that it's used to build new proteins. So the amino acids are used to build new proteins, such as enzymes. Remember that enzymes are proteins in nature, cell membranes, and muscle tissues. 1 mark for saying used to build new proteins. Another fact is that the amino acids are used to make hormones, chemical messengers in the body. Examples of hormones are insulin, progesterone, estrogen, testosterone, just to name but a few. So used to make hormones or other nitrogen-containing compounds. 1 mark for each point. Excess amino acids are deaminated, meaning they are broken down to form urea. And they are removed by a process called excretion by the kidneys. So these are the 3 things that happen to the amino acids for 3 marks. Welcome, learners. We are revising a paper from the Catholic Diocese of Karonga schools such as Chaminade Secondary School, Kaseye Girls Secondary School, St. Mary's, Karonga Secondary School, St. Ignatius of Loyola Secondary School, St. Mauritius Secondary School, Ngara CDSS, Fulirwa CDSS, St. Anne's CDSS, and just to name a few, St. Monica CDSS. Remember, we also have St. Monica Secondary School in Mangochi, but this particular one is the one in Karonga. Welcome, learners. Name the end products of the following food substances. Think these are digestion products. So when proteins are digested, what do they form? Okay, this question is coming after the one above there, talking about amino acids. And we mentioned that amino acids are the final products of protein digestion. So the end products of the digestion of proteins is the amino acids. Sorry, I think we're still looking at B. So the remaining part is used for energy or converted into glucose or fats. Now, so the digestion of proteins will lead to the formation of amino acids, which we just talked about up there, and lipids, which are also called fats. When fully digested, will form what are called fatty acids and glycerol. These are molecules, the final products of lipids, fatty acids, and glycerol. Welcome, learners. My name is Mr. Pochilola, your teacher. In case you have questions, don't hesitate to simply leave a comment in the comment section, and we'll be glad to attend to you. Welcome, learners. Number 3. Describe 3 ways how antibodies defend the body. Defend the body from infections. Remember that the body has different mechanisms to defend itself from infections or diseases. One of them is to use antibodies. They're messengers in the body. It also uses mechanisms such as the skin. It uses mechanisms such as the action of HCl in the stomach. But this particular question is asking us for the action of antibodies in defending us from infections. One of the ways by which antibodies defend the body is what is called agglutination. Agglutination involves antibodies binding to antigens on different pathogens. Remember, pathogens are disease-causing microorganisms, causing them to come together, to clump together, which makes it easier for white blood cells to locate, surround, and engulf them, swallow them up, destroy them. So strategic. So the antibodies will bind to the antigens, to the proteins on the pathogens, let's say bacteria and other disease-causing agents, put them together, clamp them together, and smash them. That's one way. In so doing, the body is defended from those antibodies. Another way is to neutralize them. neutralization. Antibodies attach themselves to toxins or harmful substances produced by pathogens and block the surface proteins of pathogens. In so doing, the body is defended. So antibodies attach to toxins or harmful substances produced by pathogens, or sometimes block the surface proteins of pathogens so that they don't work. They don't cause further damage to the body cells. So this stops, in support of neutralization, this stops them from entering or damaging body cells and makes them harmless. The third way is what is known as opsonization, also called marking, right? Whereby antibodies attach to the surface of pathogens, acting as tags. This signals to phagocytes, the white blood cells, to recognize, attach to, and destroy the pathogen quickly. These are the ways by which antibodies defend the body from infections. Know all of them so that you should be able to write all of them when required, 3 of them, or you can write any 1 or 2, depends on the wish of the examiners. Another technique is what is called precipitation, whereby making soluble antigens insoluble. So the antibodies make soluble antigens insoluble so that they can be engulfed or activated as complement proteins to destroy pathogens. So know all of them. Name the type of valve which prevents backflow of blood in the pulmonary vein, in the pulmonary artery. The type of valve. Remember that it is the valves that prevent the backflow of blood. So this in the pulmonary vein, in the pulmonary artery, is achieved by what is called semilunar valve. There are different types of valves. There is semilunar valves in the pulmonary artery, bicuspid valves in the veins, and so on. Make sure you know these kinds of valves and what they do, where they work. Mention one difference between diastolic pressure and systolic pressure. Systolic pressure, blood pressure, this is in line with blood pressure. Systolic pressure is the highest pressure in arteries, which occurs when the heart ventricles contract, reducing the space in the arteries, creating a high pressure. Systolic pressure is the highest pressure in arteries, occurs when the heart ventricles contract or pump blood out. While diastolic pressure is the lowest pressure in arteries, occurs when the heart ventricles relax and fill with blood. Put very simply, systolic is when the heart beats. Diastolic is when the heart rests between beats. That's the difference between these two. When you go for testing, to be tested, to have your BP tested, the top number is the systolic pressure and the bottom number is the diastolic pressure. And in most cases, the systolic pressure, the one on top, is bigger, is higher than the one at the bottom, diastolic pressure. What is the linking together of various processes in an organism called? The body carries out different processes in an organism. An organism carries out various processes such called 7 life processes, namely movement, which is also called locomotion, respiration, sensitivity, growth, reproduction, excretion, and nutrition. All these processes are coordinated together. Movement from one place to another, talking, and all that. The body is such, the organism is such an organism, an organized system, such that all the processes in each are properly coordinated. So this coordination, or the linking of the processes together, is what is called the coordination. So the word here is coordination. 1 mark. Name 2 systems that link the processes of the body. So for coordination to be achieved, 2 systems must work together, namely the central nervous system, the central nervous system, the peripheral nervous system, and all other nervous systems together, they work together. And what is called the endocrine or hormonal system. These 2 systems work hand in hand to ensure that the body activities are well coordinated, state the difference in the direction of the impulse traveling in an axon and a dendrite. Remember, these are parts of the spinal cord. State the difference in direction. When an impulse or signal, nerve signal, is traveling, how does the direction differ in an axon and in a dendrite? So in the dendrite, the impulse travels towards the cell body of the neuron. Whenever an impulse or signal is passing through the dendrite, it means it's traveling towards the cell body of the neuron. But when it is traveling through an axon, it means the impulse is traveling away from the cell body of the neuron. What it means is the dendrite is like an input, like an input device of an impulse to the cell body of the neuron. Or you may consider an axon as an output device of an impulse traveling from the cell body of the neuron. That's the difference between. So the key words are towards and away. So the dendrite, in the dendrite, impulse travels towards the cell body of the neuron. In the axon, it means that the impulse is getting out of the cell body, away from it. 2 marks. Welcome, learners. My name is Mr. Paul Chilola, your teacher. In case you're not sure of what we are talking about, don't hesitate to leave a comment in the comment section below there. We'll be glad to attend to you. That's what we are here to be doing. Welcome, learners. Explain why nerve impulses do not jump from one fiber to another, even though the fibers are close to one another. Although the nerve fibers are close to one another, the impulses do not necessarily just jump from from one nerve fiber to another. No, they don't do that. Right? So nerve fibers are separated by a gap which is called a synapse. That you must know. So a synapse is the gap between nerve fibers. So there's a gap between two nerve fibers, which is called a synapse. So the electrical impulse cannot just cross this gap directly. It doesn't just jump from one nerve fiber to another. No. What needs to happen instead is that it causes the release of a chemical called neurotransmitters. So the electrical impulse will cause the release of chemical substances called neurotransmitters from the end of the first fiber. These chemicals only diffuse across the gap and bind to specific receptors on the next neuron. So the signal passes only to the intended fiber and does not just jump to nearby ones. Welcome learners. We are revising a paper from Karonga Diocese, or the Catholic Diocese of Karonga, where we have high reputation schools such as Chaminade Secondary School, Kasei Girls Secondary School, St. Mary's, Karonga Secondary School. This is also a girls' school. St. Ignatius of Loyola Secondary School, St. Mauritius Secondary School. There are also CDSS there under the same diocese, namely Ngara CDSS, Furirwa CDSS, St. Anne's CDSS, and St. Monica CDSS. I hope you are taking note of these points that we are raising. In case you have feedback, don't hesitate to send us just a message to our numbers, or you can also leave a comment in the comment section. How do sensory and motor neurons differ in their functions? Remember, we have different kinds of neurons. Neurons are the nerve cells. We have sensory neurons, motor neurons, and relay neurons. So the examiner is asking us, how do sensory and motor neurons differ in their functions? Sensory neurons carry impulses from receptors, as the sense organs such as the skin, the eyes, the ears, the tongue, to the central nervous system. Central nervous system is the brain and the spinal cord. Sensory. Motor neurons, on the other hand, carry impulses from the central nervous system to muscles for action, or glands, which are called effectors, to produce a response. So that's the path, that's the difference in function between the sensory neurons and the motor neurons. So sensory neurons carry impulses, signals from the receptors. Receptors are the sense organs, such as the skin, the ears, the eyes, the tongue, to the central nervous system. The central nervous system is the brain and the spinal cord together. The motor neurons, on the other hand, carry impulses from the central nervous system to muscles for action. So by saying this, one mark. Question number 5. Figure 2 shows a structure found in a plant cell. Use it to answer the questions that follow. This is one of the parts in the plant cell. You should know the parts of the cell by their structure. So name the structure. This structure is basically the mitochondrion. Don't say mitochondria. Mitochondria means there are many of them, it's plural. So the answer here is mitochondrion because there's only one. State any one substance produced by the structure. So you need to know what mitochondrion does. The mitochondrion is where respiration takes place to make energy. One of the products of respiration is water. Another one is carbon dioxide. But since it is where energy is made, it means there is also ATP there, adenosine triphosphate. So state any one substance produced by the mitochondria. This question requires us to know to recall the process that takes place there, which is respiration. So adenosine triphosphate is one substance. Carbon dioxide, this is one of the products of respiration, even water. So any one of them is correct. State any one substance. 2 marks, I'm surprised. It's just mentioning one. Why 2 marks? That's okay. That's generous enough with marks. Would have been 1 mark. Explain any 2 adaptations of the structure to its function. What features does the mitochondria have which enables it to perform its functions? One of its adaptations is that the inner membrane is folded into what is known as cristae. These are called cristae. Is folded. This increases the surface area for reactions of aerobic respiration, allowing more enzymes to be attached and more energy to be produced efficiently. You may not have to say all these things, but at least you should say that it is highly folded to increase surface area for respiration. That's 2 marks. Another adaptation is that it has double membrane structure. There's this membrane inside and another membrane outside. The outer membrane controls the entry and exit of substances, selective, while the inner membrane houses the respiratory enzymes to keep the respiratory enzymes. This separation creates distinct compartments for different stages of respiration, making the process more organized and effective. Another one is that it contains DNA and ribosomes. It has its own DNA and ribosomes. This allows it to make its own enzymes and proteins needed for respiration, so it can function independently within the cell. Welcome, learners. So take any 2 for 4 marks. Don't write all of them, just 2 of them, because each is 1 mark. You mention the adaptation and explain how it functions. Welcome, learners. Explain how the medulla oblongata regulates breathing in the human body. Remember, the medulla oblongata is one of the parts of the brain apart from the cerebellum and cerebrum, apart from structures such as the hypothalamus and the thalamus. Explain how the medulla oblongata regulates, controls breathing in the human body. So the medulla oblongata detects changes in the carbon dioxide concentrations, but also acidity in blood. When it detects changes either low or high, when the carbon dioxide is high, it sends signals. The medulla oblongata sends signals to the breathing muscles. So this makes breathing faster and deeper. In the process, carbon dioxide is removed. So when CO2 is low, it slows breathing down. So this process is an involuntary process. You don't have to think about it. As long as carbon dioxide levels change, it sets into action. This could have been 4 marks, but that's fine, because the last point really is not quite strong. The independent point in relation to the ones above there, but that's fine. Make sure you're able to mention these things. How medulla oblongata regulates breathing in humans. It's like a sensor sensing the concentration of carbon dioxide in the blood. Explain how inhalation occurs in human beings. Remember, inhalation is breathing in and exhalation is breathing out. So explain how inhalation occurs in human beings. Alright, so a number of processes occur. One of them involves diaphragm contracting. The diaphragm contracts and moves downwards during inhalation. External intercostal muscles contract, pulling ribs up and out. In the end, the volume in the chest cavity increases. And pressure inside the lungs falls below air pressure outside, or atmospheric pressure. Then air flows into the lungs. So when we breathe in, intercostal muscles contract, pulling ribs up and out, upwards and outwards. The diaphragm contracts and moves downwards. The volume of the chest cavity increases. And as a result, pressure in the lungs decreases below air pressure outside the body. This causes air to flow into the lungs. Okay, I think these points need to be in order. All right, so when you breathe in, the first thing that has to happen is the intercostal muscles, especially the inner intercostal muscles, contract, pulling the ribs up and down. This would result into the diaphragm contracting and moving downwards. Then this is going to be followed by the increase in volume of the chest cavity. And the volume increases, pressure inside decreases. The relationship between volume and pressure, this is Boyle's law, basically. Remember that Boyle's law tells us that pressure is inversely proportional to volume at constant temperature for a fixed mass of gas. But anyway, that's just physics. It still applies here. Increased volume leads to decrease in pressure. So air flows from the outside the body into the lungs. It rushes into the lungs. Welcome, learners. Once again, let me emphasize these points are not in order. First of all, the first one, when you breathe in, intercostal muscles contract, pulling ribs and up and out, upwards and outwards. This causes the diaphragm to contract and to move downwards, thereby increasing the volume of the chest cavity, and pressure inside the lungs falls below or decreases, allowing air to rush into the lungs. 5 marks. Make sure all these points are there in order to get the 5 marks. Although the embryo depends on the mother's blood for its food and oxygen, its circulatory system is never directly connected with the maternal blood vessels. Explain how. Explain what would happen if the two circulatory systems in question were directly connected. Give 2 points. So the circulatory system of the mother and that of the embryo are not joined directly. Alright? So something wrong would happen if the two circulatory systems were joined directly. So the question is, what will happen if the two circulatory systems in question were directly connected? The mother's blood pressure could damage or destroy the delicate blood vessels of the embryo. As the embryo's circulation is much weaker, so there would be pressure difference which would damage the delicate blood vessels of the embryo. That's one thing that would go wrong. Another one is that the mother's immune cells or antibodies would enter the embryo's blood, thereby recognizing it as a foreign and attack it, causing rejection or harm to the baby. These are the two things that would go wrong if the circulatory systems of the mother and that of the embryo were connected. Welcome, learners. Another one is that the difference in blood groups could also cause dangerous clumping, which is called agglutination of the blood cells, risking death of the embryo or the mother. Either way, it's either the embryo killing the mother or the mother killing the embryo due to differences in blood groups. So if the two mixed up together, there would be dangerous clumping, which is called agglutination. But these don't happen because the two circulatory systems are not connected directly. Figure 3 shows levels of some hormones during the menstrual cycle. Use it to answer questions that follow. So we have the concentration of hormones, 2 hormones, estrogen and progesterone. This continuous curve represents estrogen, and this broken curve here represents progesterone. And then we have the time of the menstrual cycle in days. Remember that the menstrual cycle is 25 to 28 days long. During which period is fertilization likely to occur? Remember that fertilization is most likely to occur when an egg has been released in a process called ovulation. So ovulation is most likely when the concentration of estrogen is high. Just immediately after the progesterone levels are high, it will trigger the release of an egg. That means anywhere from 12 days, day 12 to day 14 or 16, they're about here when the peak of estrogen is high, because the levels of estrogen will trigger the release of an egg by ovulation. So during which period? So it is between the periods of 14 and the day 16. Here, this is not safe. Something needs to be done by all means to prevent fertilization if it's not needed. So the reason for this is that estrogen peaks just before this time, just before now, before day 14 and 16. Estrogen peaks just before. Estrogen level is before, just before this time, before this time here. That means anywhere here, 10, 11, 12, 13, the egg is released. So the egg is available during this time for fertilization, only for a short time after ovulation. There's a link between estrogen levels and the probability of releasing an egg. So the highest peak reached for estrogen levels is day 12. It's very high. That means the egg must be released between day 12 to 14. So the egg is still alive, it's still available for fertilization. State any two things that may happen to the walls of the uterus between days 5 and 10. Let's go to days 5, 10. Day 5 is here, day 10 there, between this time. Considering the levels of you can appreciate that estrogen levels are rising, while progesterone levels remain constant up to after day 12, and then slightly begins to increase, and then it picks up like that. But we are looking at this period here. What will happen? Two things that may happen during this period is that the uterine wall repairs and thickens. So the uterine wall begins to become thick again. It is generating itself during that time. And blood capillaries and glands develop in the lining. The uterus is recovering from the menstruation, from the endometriosis. Endometriosis, that's the endometriosis, the breaking down of the uterus, the uterine wall. So the uterine walls are recovering, so you expect it to be repaired and to become thick again as it has to be, and also the blood capillaries and glands develop in the lining. Explain why the level of progesterone increases from day 16 to 20. By the way, does it? Day 16 to 20. Progesterone, yes, it increases. From day 16 goes up. 20 is when it has reached its maximum level here. So the examiner is asking us why. So after ovulation, which is day 14, the empty follicle becomes what is called the corpus luteum. So the corpus luteum secretes large amounts of progesterone, so the level rises. What are auxins? Auxins, in relation to plants, are plant growth hormones, which are the substances that control growth and development. Welcome, learners. We are revising a paper from the Catholic Diocese of Karonga. Schools there include Chaminade Secondary School, Kasai Girls, St. Mary's Karonga, St. Ignatius of Loyola Secondary School, St. Mauritius Secondary School, just to name a few, and some CDSSs such as Ngala, Mulirwa, Fulirwa, St. Anne's, and St. Monica. They are the ones who wrote this paper. Very informative, very rich paper, very worthy paper, exam style paper. Welcome, learners. Define the term auxins, the plant hormones. How do auxins concentration affect growth of the foreign parts of the plant? Shoots. Remember, shoot is the part of the plant that grows above the ground, consisting of the stem, the leaves, and everything else growing above the ground is called shoot. So how does auxin concentration affect the growth of shoots? Alright, when the concentration is high, the auxins will stimulate. High concentration causes an increase in growth. So when the concentration of auxins increases, growth increases. 1 mark. The roots grow below the ground, underground. When the concentration is high, growth is inhibited. In other words, there is slow growth, or low concentration stimulates high growth, the opposite. So when the concentration of auxins is high, the roots don't grow that much. But when the concentration is low, growth is enhanced, or there is stimulated growth. Explain how nastic response occurs in the following. Mimosa pudica. Remember, Mimosa pudica, the plants that respond to touch. When you touch them, they shrink away from your touch. They are very common in Mulanje, in a place called Mimosa. I think the place is called Mimosa because they are abundant. Mimosa pudica plants there. So nastic response is the response of plants to touch. Plants moving from a stimulus, a touch stimulus, mimosa. So explain how nastic response occurs in the following. Mimosa pudica. So touching stimulates cells at the base of the leaves to lose water. So water moves away from the base of the leaves. As a result, turgor pressure decreases and the leaves fold. You may think that they are moving. That's nastic response in Mimosa pudica. In Venus flytrap are those plants that are thought to be eating insects. They don't eat per se. They just respond to the insects, the stimulus, in this case the insects landing on them. So they fold themselves, appearing to be swallowing the insects, digesting them. So touch triggers rapid water movement in cells, changes shape causing the trap to close. So the insect, such as bees, are trapped by such changes. And you may think that the insect has been swallowed or eaten. That's the notion many people have. This figure here shows a feeding relationship. of the relationship among organisms and the environment. What does F represents? So F is pointing towards producers. Producers are basically plants. So plants use sunlight to carry out photosynthesis. So F must be sunlight. F must be sunlight. If you say light energy, that's fine, or solar energy, it's okay. Because source light from the sun is also called solar energy. Explain how the plant benefits from the activities of organisms represented by the letter E. The organisms here which act on the dead plants and dead animals. Producers die. When animals which eat plants also die, and they are acted upon by organisms represented by the letter E. So how do plants benefit from that? Alright, so organisms E are basically decomposers, breaking down dead remains of producers and consumers, plants and animals, by decomposition. This is followed by the release of essential nutrients like nitrates, phosphorus, and minerals into the soil. So these are very essential to plants. So producers absorb these nutrients through their roots to make food and growth. 3 marks. Number 10. Why would a plant fail to carry out photosynthesis if it lacked magnesium? Lack of magnesium in a plant would affect photosynthesis. So the examiner is asking, how would a plant fail to photosynthesize if it lacks magnesium? Recall that magnesium is needed to make chlorophyll. So imagine a plant without chlorophyll. What would go wrong? So without chlorophyll cannot form. Without magnesium, chlorophyll cannot form. So light cannot be absorbed for photosynthesis. 1 mark. Trust me, this could have been 2 marks also, but that's okay. Welcome, learners. How does a green plant reduce carbon dioxide concentration in the atmosphere? How? We say that green plants reduce carbon dioxide concentration in the atmosphere. How does it do that? All right. So during photosynthesis, you need to be aware of the fact that plants take in carbon dioxide from the air through what is called carbon fixation, also known as carbon sequestration. So during photosynthesis, remember that carbon dioxide is needed for plants to make their own food as a raw material alongside water. So as a result, they use it to make glucose, removing it from the atmosphere and lowering its concentration in the process. Name any one chemical reaction that takes place during light stage of photosynthesis. Remember that photosynthesis occurs in 2 stages, namely the photo stage and the dark stage. So we are asked here to name any one chemical reaction that takes place during the light stage of photosynthesis. So one of the reactions is what we call photolysis of water. So photolysis is when light is used to split water into hydrogen and oxygen ions. So you can name photolysis of water as an answer. but also formation of ATP, adenosine triphosphate. It's when during this stage of photosynthesis, light energy is converted into chemical energy. So you can mention formation of ATP or photolysis of water. Even formation of ADP, adenosine diphosphate. Welcome, learners. Question number 11. We are in section B now. We thank God for bringing us this far. Welcome, learners. This is Black Slides Online Secondary School. Consider subscribing to the channel if you have not done so already. Explain 5 adaptations of the renal tubule to its function. Remember that the renal tubule is found in the kidney and is part of the nephron of the kidney. It delivers the products of excretion. So what features does it have for it to carry out its functions? So one of the adaptations of the renal tubule, that's where reabsorption of essential substances takes place, such as reabsorption of water, vitamins, proteins, even glucose and others back into the blood. So the renal tubule, I would urge you to go back to the structure of the nephron. Look at it and locate the renal tubule. It's long and coiled to give large surface area for maximum reabsorption. By saying it's long and coiled, that's 1 mark. Remember, this is an essay question. We need to back up any claims that we make. So the fact that it's long and coiled, it gives it large surface area. It increases large surface area for maximum reabsorption. That's 2 marks. The renal tubule has walls that are 1 cell thick. By saying 1 cell thick, 1 mark, to allow fast diffusion of substances, another mark. Already have 4 marks for saying this too. The inner lining has microvilli to increase surface area for absorption. Inner lining having microvilli, 1 mark, to increase surface area for absorption. 1 mark, that's 2 marks. Cells have many mitochondria, 1 mark, to provide energy for active transport. Remember that mitochondria is where respiration happens. Respiration is the energy producing reaction. So cells have many mitochondria, 1 mark, to provide energy for active transport. It's surrounded by thousands of blood capillaries, 1 mark, to carry away reabsorbed materials quickly. So 2 each multiplied by 5, that's 10 marks. Welcome, learners. My name is Mr. Paul Chilola, your teacher. Question number 5, rather 12. Describe the processes that describes that describe the Darwin's theory of evolution through natural selection. That's the issue of survival of the fittest. So it has 5 components. We have to describe those to give us 10 marks. So first and foremost, let's state the obvious. Organisms produce more offspring than can survive. That's a fact. Meaning that organisms produce many offsprings, leading to competition for resources. So the many offsprings compete for resources such as food, shelter, air, and others. Now, individuals in the species show natural variations or differences in their characteristics. Alright, so those for favorable variations, those with strong variations are better adapted to the environment and they survive. The survival of the fittest. So survivors are going to reproduce and pass on the beneficial traits to their offspring. Inheritance. So over time, favorable traits become common and the species gradually change. Which is evolution through natural selection. So take note, there are 5 points. 1, 2, 3, 4, 5. So each point has 2 marks. Take note. For every claim you make sure you back it up. Welcome, learners. Question number 13, the last one. Explain any 5 applications of biotechnology. So biotechnology has a number of applications. You need to know some of them, at least 5. Biotechnology is used in medicine by mentioning where the area in which it's used, medicine, that's 1 mark, and then explaining it. So it's used in the production of insulin, vaccines and antibiotics using genetically modified bacteria. 2 marks for saying this. It's used in agriculture, that's 1 mark, creating pest-resistant, high-yielding, genetically modified crops, another mark. Food production, 1 mark. Use fermentation to make bread, cheese, yogurt, and alcoholic drinks, another mark. It's used in waste treatment where we use microbes such as bacteria to break down sewage and clean up oil spills. Welcome, learners. Lastly, it's used in industry in the manufacture of enzymes for detergents and biofuels from plant materials. Take note that when responding to essay questions, you don't put bullets, just put paragraphs, your points in paragraph form. And it doesn't mean that you should write long paragraphs, no. Just points that make sense. Welcome, learners. My name is Mr. Pochilola, your teacher. It's been a wonderful time together. This is close to 1 hour 30 minutes of recording this revision. Remember, we were revising a paper from the Catholic Diocese of Karonga, far north. Schools from that diocese include Chaminade Secondary School, Kaseye Girls, St. Mary's, Karonga, St. Ignatius of Royora, St. Mauritius Secondary School, and some CDSS schools which are managed by the diocese, including Ngara CDSS, Fulirwa CDSS, St. Anne's CDSS, and St. Monica. For the time being, I wish you a wonderful day until we meet for the next